Dear fellows, welcome to the 7-day research course, global workshop on basics of research. And in previous days, we have studies about the importance of the research, and then we studied about the synopsis writing, and in detail just studied that how we will write and compose our synopsis. So the today lecture is about designing experiment for thesis or article research and data analysis. It's like a guide to experimental design, and whenever you have to you conduct research or any type of work in research, you have to design different type of experiments. And today we will study about the experiments, this how we can work on that type of experiments, and how we can design the experiments, and then how we can analyze the data through different softwares like SPSS, Python, and different other softwares. So today we will first study about the introduction of experimental design, and discuss in detail about the experimental design. Then we will discuss some common steps, and then detail examples from various field, and then we'll summarize our lecture. So let's come to the introduction of experimental design. What is experimental design? If you look at definition, a structured approach to planning and conducting experiment. If you are planning an experiment, or you are conducting an experiment, you have to design experiment. You have to do the experimental work, and for that work, you have to design different type of uh, experiments, and according to your need and your research. So experimental design basically involves planning and conducting studies to test hypotheses. And if you look into the medical research, it helps establish cause and effect relationship between, if you are looking into some type of doing intervention, and then you want to check its outcome, so you can do that thing through research. And experimental design basically ensures reliability and validity of the study. It also avoids biases, and it enhances the reproducibility. If someone else in some other country want to conduct that type of study, and then they can easily conduct if you have designed a good experiment. Now, experimental design is a structured framework for planning and executing studies to ensure results are reliable, valid, and reproducible. So you have to make sure that the results which will be produced after the experimental design, or based on the experimental design, must be reliable, and they must be valid, and can easily be reproducible. Now, if you study that why it matters. And if you look into the reliability and validity, or like what does reliability means, that consistency and repeatability of measurements, and validity means accuracy and relevance of measurements. So it must be reliable, and it must be valid and reproducible. And why it matters? Because it minimizes bias, like selection bias in participants groups. So bias refers to systematic errors or distortion in research findings, and common type of bias include selection bias, and like information bias, and confounding bias. So there are different type of bias we can discuss. And it also enhances the credibility for peer review. If you have designed a good experiment, then you can submit it to a very quality and high rank journals, and it can enhance the credibility for the peer review. And for example, a double blind trial, drug trial design eliminates placebo effect. So if you work on your experimental design, your results will be reliable, your results will be valid and reproducible. So after the synopsis, or even if you are writing synopses, and you are writing the methodology or experimental design part, you have to work on that thing to make sure that after working on all these things, and implementing this type of experimental design, you get the reliable and valid results which can be easily reproducible. Now, there are two types of research. If you look into the quantitative research and quantitative, basically quantitative statistics or quantitative research is the research of numbers, statistics, or objectivity. It refers to these things. And in qualitative, words, meaning, and subjectivity are discussed, and both can be rigorous and complementary. Both can be used even if you look into medical field. So quantitative research can be used to, let's say, evaluating treatment efficacy, identifying risk factors, and qualitative research can be used to understand patient experiences, exploring healthcare perceptions. and all these things. So both can be rigorous and complementary. Now, what are the difference between quantitative and qualitative research? So if we discuss these things in detail, basically in quantitative research, the data type is in numerical, for example, blood pressure readings, and in qualitative research, the data type is descriptive, like patient interviews, patient surveys, and the analysis which is done for the quantitative study is statistical analysis like t-test, ANOVA, chi-square, and all the things. And for qualitative research, you have to do thematic coding and narrative analysis. And the focus of quantitative research is on what and how much, for example, drug and dosage. And the qualitative research focus on why and how, like questions, for example, patient experiences. And if you look at the examples of quantitative research, let's say measuring vaccine efficacy in 1000 patients, and qualitative research exploring barriers to mental health care access. So these are the types of quantitative and qualitative research. So you have to define that what type of experimental design you need. Either you need a quantitative experimental design, or either you need a qualitative experimental design. Then you have to work on your data according to the experimental design you need for your studies. Now, the next thing is identifying the research question. You have to identify the research question in order to align with the with your study and to easily complete your study. So, basically, what is research question? If you look into these things, research question is a specific inquiry that some guides the study, and it should be clear, focused, and concise. And it also help define the scope, objectivity, and objectives and direction of the research. Basically, your research question do clear and define the scope and objectives of your research and direction of the research. And types of, if we discuss the types of research question, there are descriptive types of research question which describe characteristics or some type of some sort of phenomena. And then comparative research question which use comparing groups or interventions. And then causal research question examining cause and effects relationship. Exploratory type of research questions in which we explore new ideas or phenomena. And evaluative type of research question in which assess the effectiveness of interventions. So all these are the different types of research questions. You can use anyone according to your research and according to your study you want to conduct. And if we discuss that why research question is important and why we consider research question an important factor of research. So basically, research question provides clarity and focus. It will provide clarity and focus to the study. After forming a research question, you will be clear in your on your research, that what you want to study and what you want to achieve, and you will be focused on your study, because your research question will guide you through all this work. And it also guides you during literature review and methodology. You have to focus on the literature which is related to your research questions. And while working on methodology, you have to do consider that things, that research question, and make your methodology according to the research question in the methodology which will answer those research questions. So you have to, it will guide you through literature review and methodology. And research questions also determine data collection and analysis techniques. Either research question, your research question, if your research question say that this study will be qualitative or quantitative analysis of some specific disease, let's say malaria. If you have if your research question contains terms like you want to do a quantitative survey of malaria and its demographic, socio-demographic factors, and all these things. So your research question will guide you that how your data will be collected. You know that if you want to do quantitative study, you have to collect data either a secondary from secondary sources like hospitals or healthcare centers, or either from primary source directly. You can collect data from patients and different peoples. And then it will also explain that how your data will be analyzed. Your research question explains that how your research will be conducted, and how the data will be collected, and then how you will analyze this data. And it also ensure the research is relevant and feasible. You can easily conduct that type of research. So research questions are important for all these reasons and many other reasons. If you look at the characteristics of a good research question, what type of research question will be called as good research question? Some of you may wonder that what are the characteristics of good research question? So basically, good research question will be clear, it will be easily understood and specific, it will be focused, it will narrow enough to be answered within the study scope, and it will be feasible. It will be feasible and can be researched with available resources. It will be easily completed with the available resources. If it is significant, it will do contribute to the existing knowledge, to the scientific knowledge, and to the scientific family by contributing its results, and then the research must meet ethical research standards. It must be approved from ethical sources. And if we look at the types of research, like we have studied that there are different type of research questions, like descriptive. For example, we studied in detail that descriptive research questions basically describe some type of characteristics or phenomena. For example, what are the characteristics of XYZ or whatever you made. And then comparative research question, in which we studied that comparing groups or interventions. So for example, there is a comparative question, comparative research question, that how does X compare to Y, or how does X affect Y. So type of this question will be considered as comparative questions. And causal, what is the effect of X on Y. These are the questions which examine cause and effect relationship. And then exploratory type of research question, we discussed that exploratory research question explore new ideas or phenomena. For example, what factor influence, let's say, diabetes or some other disease or some other condition, and then evaluate you that assessing the effectiveness of some type of intervention, how effectiveness is COVID vaccine, how effectiveness is diabetes type some medicine, and all these things. So these are the type of research question, and you must focus on these things, that what are the types, what type of research question you want to use, and what type of research question will help you to achieve your goal. So this is the main factor you should work on while working on research question. And then what are the steps to identify research question? How you can identify research question? You have to choose a broad topic and identify an area of interest, and then conduct background research. You have to review the existing literature and then narrow the topic, focus on some specific issue by finding through research gaps, and then identify the research gaps, find the unanswered questions, and make sure that which of them you can answer, and then formulate your question according to all the work you have done previously. So this is the basic criteria of formulating or identifying your research questions, that you must have knowledge about your research. And then examples of formulating a research question, if you look at this, let's say broad topic is human disease, and if you narrow down the topic leishmaniasis in humans, and if we look at the research gap, lack of prevalence data of some specific disease, let's say we say that research question is how much prevalence vary among different districts. So it is type of comparative research question. So this is the way in which you can do work on your research questions and formulate your research question easily. What are the common mistakes to avoid while working on your research question? First of all, do not choose too broad or too narrow topic, and do not formulate such question which lack clarity or specificity, and do not go for those question which are not researchable with available methods, and those which lack relevance or significance. So you have to look at these things, that the question you are working on, the question you are formulating uh, is good or not good enough to be worked on further. And you can use the simple criteria which is used called as finer criteria. So for your research question, you have to use that type of criteria which is called as finer criteria. Like, your research questions must be feasible, it must be interesting and novel, and it must present something new, and then ethical, it must be achieved under ethical considerations, and it must be relevant to the research and to the scientific knowledge. So you have to use the finer criteria. After that, we will study about how to conduct a literature review, or how you can work on literature review. So you have to define the key terms, identify essential keywords related to your topic. You want to do work on cardiology or CVDs, cardiovascular diseases. You have to first define your topics, your essential keywords related to your topic, and then you have to search different academic databases. You have to use Google Scholar, PubMed, Scopus, and other sources to find the related articles, related data, and then review the previous research and analyze existing studies to understand the trends, the gaps, and the methodologies which were used in that previous research. And then after that you have to assess the credibility. For that purpose, if you are studying articles, you are using data from some articles, you have to make sure that you prioritize peer-reviewed journals and reputable sources so that your research work does not get some issues in later stage. So always get data from the articles which are published in peer-reviewed journals and reputable journals. So then after all these things, you have to define the keywords, then search databases, then conduct, then you collected data and then studied that thing. And then yeah, now you have to organize findings, summarize the key points and categorize information for clarity, and then identify research gaps, that what are the unanswered questions which you can answer, and which can be easily refine your research focus. So you have to do this criteria, to follow this criteria for literature review. Now the next thing in this lecture is formulating a hypothesis. For every research, you have to formulate a hypothesis. You have to make a hypothesis which is good enough to be so that you can easily work on that hypothesis and then prove it true or false or whatever may be the condition, but you can easily work. So if you look at the definition of hypothesis, so hypothesis is a testable statement that predicts a relationship between variable based on existing knowledge. So it is the testable statement that can be tested. Hypothesis can be easily tested, and it predicts a relationship between some sort of variable, and that relationship is based on the existing knowledge. So this is called as hypothesis. And if you look at the type of hypothesis, usually we use two types of hypotheses. The first one is null hypothesis, which we call HO or H naught, states that there is no effect or relationship between variable. Let's say there are variable A and variable B, and we say that the null hypothesis will be that there is no effect or relationship between variable A and variable B. So null hypothesis basically show no significant difference or relationship between variables, and there is alternative hypothesis. Alternative hypothesis basically show significant difference or relationship between variables, and it suggests that there is an effect or relationship between some sort of variable. So these are the two types of hypotheses. And if we look at the characteristics of good hypothesis, a good hypothesis is clear and specific. You have to avoid vague and or hourly broad statement. And a good hypothesis must be testable and can easily be malleable using experiments or observation. And it should be based on existing knowledge and must be rooted in prior research and theories. And it must be logical and rational and should make sense given the available data. So these are the things you have to keep in mind while working on your hypothesis. And then what are the steps to formulate a hypothesis, to work on your hypothesis or to write your hypothesis? So first of all, you have to identify the research question and clearly define that what you want to study, what type of study you want to do. And after that thing, you have to formulate the research question and work on your research question. And after completion of your research question, you have to then work on the review literature. You have to review different type of literature and gather background information which will support your prediction, which will support your research question, and then define the variables, that what variables are independent and what type of variable will be dependent in this study. So you have to do explain these independent and dependent variables, and make a prediction. Now after all these things, you have identified research question, you have done literature review, clearly mentioned your objectives, and you have defined variables, and then you have to make a prediction. This prediction is called as hypothesis, and this prediction is to formulate a statement that will describe the expected relationship that you want to study. That type, like in everyone in class grade 9 or class 9, you all have studied, or in school we have studied that there was a hypothesis about malaria, that Anopheles mosquito cause malaria. That was a hypothesis. So you have to make that type of statement that will describe the expected relationship between two variables or many variables, and then ensure testability. And you have to confirm that the hypothesis can be verified through experiments or observation, and hypothesis must be testable. So you cannot make a hypothesis that is not supposed to be tested in any type of situations, and you say that this is my hypothesis. So you cannot make such type of hypothesis. And then let's say if we study, for example, research question, and your hypothesis will be based on your research question. So research question is very important, and your whole research revolves around your research questions. So let's say this is the research question, that does caffeine improve memory performance? Now hypothesis. Now that there is a two type of hypotheses which we studied, that one is the alternative hypothesis, and second one is null hypothesis. So the alternative hypothesis is that individual who consume caffeine will have better memory recall compared to those who do not. So it do verify, or it do support that there is some sort of relationship between the variables, and says that any the people or the individuals who consume coffee have a better memory recall compared to those who do not use coffee. So this is alternative hypothesis, and the null hypothesis will be that caffeine has no effect on memory recall. Caffeine does not affect your memory. Caffeine does not recall your memory. So this is null hypothesis. So basically, these both hypotheses are used. One is alternative hypothesis, in which you confirm that there is a relationship between two variables, and the second one is null hypothesis, in which you deny the relationship, that there is no relationship between the variables. So you have to make both hypotheses. And then if we look at the criteria for a strong hypothesis, what is the criteria for a strong hypothesis? And so the first thing is that strong hypothesis must be testable, and it can be supported or refuted through experiments. Like you cannot refute or support some type of hypothesis without any experiments or without results. So it must be testable and can easily be supported or refuted through experiments. And for example, let's say that daily meditation reduce anxiety score by 20% in college student. So this is type of hypothesis. This is hypothesis, and you can easily test this hypothesis if you want to see that does daily meditation reduce anxiety score. So you have to conduct a survey in college to with college students, and then you can easily do analyze this hypothesis after collecting data from college students and analyzing that. You can find out that either this hypothesis is true or not true or false. So the second thing criteria is your a strong hypothesis must be clear and specific. It avoid vague terms like might affect. For example, weak hypothesis is that social media impacts mental health. So this type of weak hypothesis, because it does not show that how much time spent on social media will impact mental health, and how much mental health will be affected from these things. So this is type of weak hypothesis. If you look at the strong hypothesis, that let's say 3+ hours of social media daily increase depression risk by 30% in teens. So this is type of valid hypothesis, strong hypothesis. You can easily conduct study on this hypothesis by in people who use 3+ hours on social media. So you can easily conduct a survey on this thing. So this is clear and specific. It must be theoretically grounded and must align with the existing literature. For example, building on prior studies linking sleep deprivation to cognitive decline. That there are previous studies that deprivation is cause cognitive decline, and you want to conduct study on this thing. So there must be some sort of knowledge behind that hypothesis. There must be a theoretical ground for that hypothesis, and you have to search for that thing and mention these things in your literature review. Now, while choosing the sorry, while choosing the experimental design, so experimental design, like we studied earlier, that it is a framework or plane which is used to conduct an experiment systematically to test some sort of specific hypothesis. What are the key considerations in experimental design? So first of all, the key consideration is the objective of the study. Objective of the study determine whether the experiment aims to establish causation, correlation, or description, and your objective will define the type of experimental design, that what type of your objectives are, and your experimental design will be based on the types of the objective you have used in your study, and the types of variable you have to identify the independent, dependent, and control variables. And then you have to find a research experimental design to work on experimental design which is feasible and can be completed in considerable time, resources, and have no ethical constraints. And for example, if you look at the type of experimental design, descriptive design, we have studied that it observe and regard phenomena without intervention of anything. It's just description and observation. And the second one is correlational design, which examines the relationship between variable but does not establish causation. So it just like we have studied in our SPSS course, and we will again study these things, correlation. So correlation does not means causation. So basically, this is the type of experimental design. You can use that type of experimental design. And the next one is experimental design, true experiment, like in which we use randomization and control randomized trials and in control conditions to test causal relationships. So we use these things to work on different type of experiments in which, like, we study randomized control trials or different type of clinical trials. And the last one is Hossi experimental design. It's often used in research and academia to describe something that is almost but not quite a certain thing. For example, Hossi experimental design, which is similar to a true experiment, but it do lacks full randomization. So, what if you're choosing between experimental design? What type of experimental design will you choose, and based on what type of characteristics? So, first of all, common experimental design methods are between subject design, that there different group receive different conditions, and you are studying the you are working on between subjects design, and you can also use the experimental design within subjects. The same participant experience all condition, and the factorial design, in which you investigate multiple factors and their interaction with each other. And the last one is control and experimental groups, which ensures comparison between treated and untreated groups. And what are the steps to choose an experimental design? First of all, you have to define the research question and hypothesis, define the variables and how they will be measured, and then select the most suitable design based on your study objective. And you have to ensure that proper randomization. You have to make sure that the data is randomized and control to reduce bias, and plain data collection and statistical analysis methods. So, for example, there is a research question that does COVID vaccine improve immunity? So experimental design will be randomized control trial with one group receiving the vaccine and another group receiving no treatment. So this is the way you can easily conduct your research and work on your research. So first of all, you have to make sure that what type of study you want to conduct, and what are your research questions, what are the hypotheses, and what are the objectives of the study. So you have to make sure that you keep in mind all these things. And there are key principles while choosing the experimental design. First one is the validity. You have to ensure the results reflect reality. For example, control group in drug trials, you have to make sure that they are valid, and there is validity and reproducibility. Detailed protocols are all protocols are explained in detail to make sure that it can be easily reproducible in any part of the world, and efficient. That the experimental design is balanced cost. It is not too much expensive, and it is time cost and resources. So you have to look at all these things. And the common designs are randomized control trials, RCT, which are gold standard for clinical study, and then cohort study, which track groups over time. For example, smoking versus non-smoker, and case control study, which compare cases disease with control where no disease. So these are the types of common designs which are normally used in medical field. If you look at the medical field, there most of the studies are randomized control trials and cohort study. So all these things are getting used. So there now, if you look at the variables, that how you will set your variables and what are the type of variables. So the independent variables are the variables that are being manipulated or tested, and the dependent variable are the outcomes or response which is being measured, and control variable are the factors that are kept constant to minimize biasness. And there, if you look at the types of variables, there are independent variables, which are the variables which is being manipulated or tested. For example, drug dosage, and dependent variable, which are the measured outcome. For example, pain reduction. And then confounding variables, which are external factors to control, for example, age, diet, etc. And the example of study effect of caffeine on memory, independent variable is here is caffeine. Its intake is 0 milligram, 15 milligram, and 100 milligram. 0 mg is the control group, and 50 and 100 are two groups, experimental groups. And dependent variable is recall test scores, and confounders are sleep duration, stress level. So you have to look at all these things. Now, how to select a sample and sample size? You have to use statistical methods for advocacy and ensure the randomization to avoid biasness, and make sure that the sample selection is random sampling, and it will reduce selection bias. For example, lottery system, if you look at the random sampling, random sampling are type of sampling in which participants are selected randomly, and the second one is stratified sampling, which ensures subgroup presentation. If, for example, equal gender ratio, and stratified samplings are participants are selected based on specific characteristic, for example, based on their sex, gender, and any other thing. So sample size, you can use tools like G-Power or formulas to determine minimum participants. For example, a drug trial may require 200 participants for 80% statistical power. So you have to use different type of models to train your data, to work on your sample size and all these things, and sampling data collection and all these things. So how to implement the experiment? You have to follow a predefined protocol which you have completely written down while writing your synopsis, and then make sure that consistency is present in data collection. And then the checklist is to pilot test to identify flaws. If you have make a research question and you want to test and you want to conduct to collect data through that questionnaire, first of all, do a pilot test, fill it from 10 to 15%, and then analyze it, study it, and then look for the flaws. If there is some sort of issues, you can change that it easily. And then a standardized procedure, for example, same lab condition, or train staff to minimize human error, and document every step for transparency. For example, in the plant growth study, use identical pot, soil, and light exposure. So you can use these things to make sure that your experiment is completely implemented and is working 100%. So for data collection and data analysis, you can use tools like SPSS or Python, and you can apply statistical tests. We will discuss in detail about the statistical test in our next lecture. So how you can collect and analyze data? If you can collect quantitative data and analyze quantitative data through SPSS, R, or Excel, and different type of tests like regression, chi-square, and p-values are measured, and then type of qualitative analysis in which NVivo and max QDA and different methods, coding themes, and sentiment analysis are done. And then visualization, you can use bar charts for good comparison, heat maps for pattern in large data sets. So you can use all these things to make sure that your results are well visualized, your results are well compared, and it's in good form and can easily be represented in any research conferences or seminars. And now let's come to the drawing conclusion and discussion that you have to relate results to your hypotheses, and discuss the limitations and the future research which can be done in that field. And writing a conclusion and discussions, you have to link results to the hypotheses, discuss the limitation, for example, small sample size or any other sort of limitation which may have, and then suggest the future research. For example, you say that the longitudinal study must be done, and you have to avoid our generalization. Like if you say that this proves the all patient need X or Y or some type of other thing. So you cannot ignore conflicting data. And for example, when caffeine improved memory in young adults, further study are needed on early elderly population. So you have to make that way to the future researcher to conduct it further and to analyze it further. So if we study different examples from different fields, let's say if we study from life sciences, is drug efficacy study. So the research question is that does drug X reduce blood pressure? Hypothesis is which is alternative hypothesis. We usually write alternative hypotheses in our research and our articles and theses. That hypothesis is that drug X lowers blood pressure significantly, which do says that there is some sort of relationship between these two. And the null hypothesis will be that drug X does not lower blood pressure significantly. So the design will be randomized control trial, and the sample will be 200 patient, and the analysis will be t-test for statistical significance. Now, if you look at the case study, testing a new antidepressant, for example, design will be double blind RCT with placebo and you can get 300 adults with moderate depression, and the variables will be independent variable will be drug 15 milligram per day versus placebo, and dependent variable will be depression score. Let's say we can analyze it through PHQ-9 scale, which is used, which is a common scale used for depression score. And let's say the outcome is that drug group showed 40% lower PHQ-9 score, and the p-value was significant. And then if you look at the natural science plant growth, it is easy that how does sunlight affect a plant height, and hypothesis that increased sunlight promotes growth, and design will be controlled greenhouse experiment. 100 plants will be selected and ANOVA to compare their growth rates. And then the natural sciences thing, if you look at these things, that impact of fertilizer on wheat yield, yes, control greenhouse experiment can be done in these things. And then the group A will receive organic fertilizer, group B will receive chemical fertilizer, and control will receive no fertilizer. And the outcome may be that group B have 25% higher yield, p value is significant. So this is how you can use different type of this data and this experimental design and things in different fields. And then if you look at the social study like consumer behavior study, for example, research question is that do personalized email increase sales? Hypothesis will be that yes, personalized email boost conversation, and the designing A/B testing and the sample of 1000 participants, and you can do analysis through chi-square test. So case studies effect of packaging on snack purchases, and survey with 500 participants. Independent variables were packaging colors, red versus blue, and dependent variable were purchase intent 1 to 10 scale. And outcome was that red packaging increased intent by 50%, and p-value is significant. So let's conclude this study and this lecture. Experimental design basically ensure reliable research, and it provides structured approach to minimize bias, and it's also provide different methods fit different for research needs. Different methods which are suitable for different research needs, and the practical application which will reinforce its importance. So we have studied all of these things. We have discussed in detail about the proposed design, which will ensure the credible results and clearly defined variables and control co-founders, and match analysis method to data type and different type of qualitative and quantitative study. So you can use tools like consort flow diagram to visualize experimental steps. And this was the end of this lecture. If you have any question, you can easily ask here in chat, or you can even send us your question directly.